Hey, if you didn't get a chance to mark that, mark the attachment sheet, and then get started. Thank you. Okay, we'll go ahead and get started now. This is AP1 lab. This is going to be the Tuesday, Wednesday, and Thursday labs. 1 o'clock in the afternoon are all AP1 labs. They're all the same. If you need to attend if you miss a lab for example, if you miss on Tuesday, you can make it up on Wednesday or Thursday, depending on your schedule. But we have 3 labs in a row that are exactly the same. That will probably become a little more important to you as we get closer to lab exam times. So, because if you're not quite ready to take your lab exam on Tuesday, you can take it on Wednesday or Thursday. So I'm the only one doing A&P here, and I'm not real picky about if you have to change your schedule around. Yeah. I was a student once too, you know, so I understand now. So AP1 lab, 1 credit course, it does not have any effect. First thing, I'll throw this out right away. It has no effect on your admission into any of the healthcare programs. It has no impact. You know, the only impact it has at all is on your overall GPA. Remember, it's a 1 credit course, so it doesn't have a whole lot of impact there either. So no one in any of the healthcare programs is going to get real excited if you got an A in lab and a C in lecture. That's not going to help. So if it comes down to making a choice between getting an A in lab or an A in lecture, get the A in lecture. B in lab is not going to hurt you. You know, it's going to help your GPA a little bit. An A is going to help your GPA a little bit, but it's not going to have a big impact. So I guess what I'm saying is, you know, focus on lecture. Do a good job in lab, but focus on lecture. That's the important thing. So, and I didn't get a chance to say this in either of the 2 AP classes that met this week. But if you're going into healthcare, then the A&P lecture is your most important class. Don't let anybody else tell you something differently. Don't let anybody tell you that this history class or this English class or this psychology class is absolutely essential to you. I mean, yeah, you need to get a good grade in the course, in those other courses, and you can't just blow them off. But the class that counts for what you want to go into is your A&P class. So, you know, it's not going to get, you're not going to get into the nursing program with an A in psychology and a C in anatomy. Let's tell you that right now. You know, nobody, no matter what it says in the catalog, nobody gets in with a C. I can tell you that right now. People get in with B's, lots of people get in with A's, but nobody gets in with a C. And, you know, it'd be nice, you know, if you were Dr. Rollinson for psychology and you get an A from Dr. Rollinson and a C from me, that's nice, but it isn't going to help. So come on in. So I want to work for attendance. Anyway, that's my little spiel about that. AP1 lab is going to try to complement what we address in lecture, and it will track a little bit on that. Today we're going to take a look at the microscope, and we're going to look at what we call epithelial tissues, and then we may go on into muscles and nerves too. Next week, we're going to take a look at cell division and mitosis, and do some wet lab work with diffusion and osmosis. And I was going to do the microscope lab today where you actually scrape your cheek for cheek cells. But I thought, you know, if I do the diffusion lab takes about an hour and a half to run. And during that time, we'd have plenty of time to do the cheek cell lab. So I'm going to just push that off for a week. You want to know the honest truth, I didn't get a chance to set it up because Dr. Malik took up all the time in here and I couldn't get to go back and forth. That's the absolute honest truth. So, but it'll work out okay because I can set it up for next week easier anyway. So, okay, AP1 lab, we have 4 lab exams. They're not cumulative. There is no final exam in lab. I'll say that today, and I'll probably say it again in November because everybody forgets. But there is no lab exam. If you're taking this on, if you're Zooming this, you can take your lab exam online. If you're on ground, you know, and you can't get in, you can take it online. You know, the lab exams are going to be strictly based on what we talk about in lab with the PowerPoints. The lab lecture, you know, what we discuss is all found in eLearn in the eLearn content section. I have everything broken out week by week. The dates for the exams are set in concrete. I don't think I said this in lecture today, and I'll probably repeat myself again on tomorrow and on Thursday, but I don't have dates set for the lecture exams because I don't know when I want to get through a particular chapter. I have an idea, but I don't like to set a date because what if we get, you know, if we fall behind or get ahead. So what I like to do in lecture is when we finish a topic, finish a unit, then I'll set the date a week out from that and then go on to new stuff. In here, because the labs run right after each other, you know, Tuesday, Wednesday, and Thursday, we have a pretty set time frame to get everything done. We can't, you know, I have to cover everything in that time frame, so the dates are set. Plus, we have a lot of material to cover in a little bit of time. In AP1 lab, we will get done the last week of class. In AP2 lab, you'll get done early. In fact, I can tell you the AP2 lab for this fall, they'll be done by the Monday before Thanksgiving. So, and then they're done. So it's one of the advantages of the AP2 lab is that you do get done sooner. Okay, so there's 4 exams in here, 4 lab exams. Each one's worth 100 points, total of 400 points for the course. And so if you want to get an A, you need to get 360 points out of 400. That's 90%. And you know, if you end up getting a B, it's 80. It's like 320 points. So an A or B is a good grade in lab. You know, so it's all point based. So, and there's no extra credit or anything like that. Everything is all spelled out for you. There is no book to buy. We simply use the eLearn content that I have available for you. And let's see what else. We meet once a week. If you have to have, you know, most days eating and drinking here is fine. I don't have any issues with that. There are only a handful of labs that we will be doing a wet lab where we don't need to have anything to eat or drink in the classroom. When we do brain and eyeball dissection towards the end of the semester, which is a lot of fun, trust me, then obviously those days you're not going to want to be eating or drinking anything then. In AP2, we usually do, we usually manage to get some cow hearts and lungs. We dissect those. And so that's a pretty messy lab. So we try not to eat and drink during those either. So, because they come fresh. So, so there's a slaughterhouse in Morristown that gets me the parts. So so you probably don't want to be eating and drinking when you're cutting up a fresh heart too. So. But that's something to look forward to for the spring. That's right. This is still fall. Okay. Let's see. Fire alarm goes off. We go out into the soccer field. Let's see. Syllabus is in the eLearn content. You know, it lists everything. Lists the sequence we're going to follow. It doesn't list the schedule. The schedule is posted in eLearn. In the content section. It's also on the bulletin board there, and it's also at the bulletin board at the other end of the hall outside the chemistry labs. Those are the only bulletin boards we get to have access to. And let's see, there is one thing I want to try to show you here, if I can get this to come up. Let's see if this is going to work or not. See if this is working. eLearn. Okay. There's our lab. Now in the content section, you're going to see, there's the syllabus right there. And even though it says spring, it's still the correct syllabus. And that lists everything that you need to understand in the class. We also have something called the lab manual. And we really did have a lab manual at one point. We keep it in eLearn content. And what the lab manual does is it's about, I don't know, 30 pages or so. And it lists all the exercises we do and goes through things that you need to remember. So if you want a quick review sheet of what's going on, for example, we're looking at epithelial cells today. And this summarizes here, we would start out with microscope in the cell, and it gives you the learning outcomes like explain the uses of a light microscope or identify the parts. You will need to remember the parts of the microscope of that. We go into bones. It lists all the bones in here. We get into tissues, things like bones. It lists all the types of all the bone names that you'll need to know for our bone test. There are 206 bone names, and we have them all listed right here. So anyway, if you define the lab manual, and as you hear me reference the lab manual, I'm talking about this thing here. We used to actually sell these. We don't anymore. So you can go right into the content, go to lab manuals, and then right below the lab manual is the assignment schedule for fall, for Severo. There it is. So like today, we're doing the microscope. We're going to do wet mounts next week, and we're going to do tissue types and maybe get to the skin today. So we'll see. So that's how you find your way around here. If you want to look at the content, for example, go over to course information, click on content, and come down to unit 1. And right there we see the microscope. There's a PDF version and there is the actual PowerPoints. If you click on the PDF version, you'll bring up the handout format where you have 4 PowerPoint slides per page. Just like in lecture, you can print this out and use this to take notes on it. This is just a summary of everything that you would, these are the PowerPoints essentially. If you want to print out the PowerPoints individually, you can download the PowerPoints themselves and print them out, or you can download them and put them in, on a flash drive. And because if there's animations, you're not going to see it on the PDF. So we have this, I have it set up with both the PowerPoint and the microscope. Then epithelial tissues, muscles, connective tissue and nerves, and then the skin. There's always a PowerPoint and a PDF file for each. So this is all unit 1 that we're going to go over in the next 2 weeks. Next week we'll be looking at the cell and mitosis, and there's a little summary thing here on cell division, and then something on membrane transport and permeability. So some of it's real interesting, some of it's iffy. I'll just tell you that. So I'll try to make it as interesting as I can. It will get better as we go along, though. So, any questions on any of this? Am I going too fast? Am I going too slow? Should I just leave? So, okay, let's. So this is what we're going to be looking at first. The under assessments, there's nothing here, I don't think. Well, those are the old lab tests, so we won't have to move those out of there. So oh wait, no, these are the, yeah. they're not open yet, so that's okay. So there are no practice exams in lab, I'll tell you that. So, okay, let me get out of this. And see if we can get this to open up and take a look at the microscope. Everybody online should still see the slide that says the compound microscope in the cell, I hope. And I'm going to open that up. Okay. Now, I will tell you that there are very few opportunities to use a microscope in AP1 or AP2. In microbiology, you'll use the microscope a lot. In healthcare, in whatever profession you get into with healthcare, you may or may not be using a microscope, but it doesn't hurt to know your way around the microscope so you can tell when you're looking at a slide or a picture of a slide, you at least have an idea of what you're seeing. Now, remember this one point. When you're looking at an image under the microscope, there is no up or down. There is no, up or down and left and right are arbitrary because the images are usually backwards and upside down to begin with. What you're really looking at are the structures. So cells don't care if they're upside down or left or right. So, we just want to look at the structures. Microscopes allow us to do this. Microscopes allow us to look at cells, to look at tissues, to look at pathogens. We can see bacteria even with our microscopes here. The microscopes we have back there along the wall that you're going to use next week are student scopes. They magnify up to 1,000 times. We'll never go above 400 magnifications, but at 1,000 times you can see bacteria very nicely. We can't see viruses. We can definitely see bacteria. We can see tissues and we can see cells very, very easily with these microscopes. So we use them to get an idea of what the various tissues look like in our bodies. So because we're dealing with epithelial cells. Those are the coverings on the body. We're dealing with muscles. We're dealing with bone. We're dealing with nerves. We want to be able to tell what kind of tissues we're dealing with. Now, if we wanted to see great detail, this microscope is just fine for us. Even the microscopes used in microbiology are no more powerful than what we have in here. They can't magnify more than 1,000 times either. They're just a little easier to use and more precise, a little sharper in their images. So we call this this is what's known as a bright field microscope. It uses a light source and it uses a couple of lenses to carry the image to the eyepiece. If we're looking at phase contrast, we would look at something looking at structures within the cell. And we probably, that's not what these are. Electron microscopes are very specialized structures that let you see down to the level of an atom. You can see atoms with an electron microscope. You probably, have you ever seen any of these pictures of, like they show you a picture of a bed bug or something in an article on bed bugs, and it looks like something, some prehistoric monster, because of its shape and everything. And they're very, very tiny. But when you magnify them up and see what they really look like they're going to eat your leg off or something. So electron microscopes can magnify up to 100,000 times, which is amazing, with great clarity too. So, but the nearest electron microscope is probably at UT. So, but this is a bright field compound microscope. That's what these things are. Compound bright field microscope. We call it a compound microscope because it has a series of lenses stacked together. It has Early microscopes consist of usually one lens at one end of a long tube made out of brass or copper, and that's all it did. Yeah, you might have you would hold this tube and look through it and look at the image on at the other end. A compound microscope uses a series of lenses. You know, for example, this has an ocular, an eyepiece, and there's a lens inside the ocular. And the ocular, you know, it says it magnifies things 10 times. It has another lens inside these structures here. These things that are called objective lenses. The objective lenses also have a lens inside there, a very, very tiny lens. And we refer to the whole microscope as a compound microscope because it has a series of stacked lenses, one on top of another. Essentially, even though we're sending light, bending it around a little bit. So we have basically two lenses, an objective lens and an ocular. The objective lens is going to be placed directly over the microscope slide, and the ocular is what you look through in here. Yeah. These bright field microscopes depend on a light source. Now, these all have lights built in. It wasn't that long ago that microscopes like this would have a separate light source that plugged in and would shine onto a mirror. There would be a mirror, and you can see on this microscope, and I'll bring the picture up here in a second, you can see that the light source is right here. This little black circular thing is a light bulb inside there. Let's go back 1960, 1970, microscopes still had mirrors on them, and you would have a high-powered lamp source that would shine onto the mirror, and that would shine up into the microscope. It didn't have a built-in light. Many of those microscopes also were positioned straight up and down. They weren't at a convenient angle, because I will tell you that if you were a scientist in the 1950s and you used a microscope, you would spend a lot of your time bent over looking into the microscope. I imagine it was pretty uncomfortable. You know, when you consider one of the big technological breakthroughs of the 1940s and 50s was to have a microscope that you could actually tilt so you could look at it at an angle, you know, not like this where it's all already pre-shaped, but you had like a giant hinge on the microscope. So we're the recipients of some, you know, pretty good technology at this point, even though they're nothing special. I can tell you that. I mean, they're good, but they're nothing, you know, they're not extraordinary, but they work for us. And in most labs around the country, you'd have something like this, maybe a little better, a little more precise, but nothing, you know, I mean, the technology is pretty much still the same. When you're looking through the microscope, we have four lenses, what we call four objective lenses. Now, the objective lenses are going to have different magnifications. We have the first objective lens is called the scanning lens. It has a red band on it, and it says 4x. That means it magnifies 4 times. We always start out on the scanning lens at the lowest power. The next low power is actually going to have a yellow band around it. It's going to say 10x. So it magnifies an image 10 times. Scanning magnifies it 4 times. Low power magnifies 10 times. High power has a blue band and it magnifies things 40 times. So we have 4 magnifications, 10 magnifications, or 40 magnifications. Now, if you have one with a white band on it, the white band will have 100x. And that's what's known as an oil immersion lens. You have to put a little drop of specialized oil on the microscope slide and lower the lens down into that so you can see your image. At 1000 magnifications, the actual movement of the air will distort the light rays. So we have to run it through a bubble of oil. So this gives us four magnifications, 10 magnifications, 40 magnifications, or 100 magnifications. The ocular is always 10x, 10 times, 10 magnifications. So the ocular is set at 10. So if you want to know what the total power, total strength is, you multiply the objective lens value times the ocular. So a 4x, the scanning lens is going to magnify things 40 times. 10 times for the ocular, 4 times for the objective lens, or 40 times. Low power is 100, high power is 400, and the oil immersion goes up to 1,000 because 10 times 100 is 1,000. Okay. So we have the 40, 100, 400, and 1,000. In A&P, we don't use the oil immersion. There's no reason to, because everything we want to see, we can see from 40 to 400. In some places, 400 is too much. In other places, 400 is just not enough. So now the light that comes through from our light source here has to pass through a structure known as an iris diaphragm. It functions very much like the iris of our eye. The iris of our eye is the pigmented portion. Now, this iris isn't pigmented, but what the iris does in our eyes is it opens and shuts based on the amount of light coming through. If the light is real intense, our pupils get real small so that we don't have too much light hitting our retina. If the light is dim, like if it was dark, if I turned the lights out and closed the blinds, then our irises would open up and try and let more light in so we could still see. Well, with the iris diaphragm, we control the size of the opening because sometimes less is better. Sometimes we don't want to have a lot of bright light because it washes out the tiny structures we want to look at. Sometimes we want to have it fairly dim. And so we open and shut the iris diaphragm so we can get a better image. And of course, there's always focusing. We have fine focus and coarse focus. The fine focus is a very small dial on the side. It turns rapidly. This is the fine focus here on the inside dial. The outside dial is harder to turn. That is the coarse adjustment. Usually what happens when you are looking at an object under the microscope is you turn the knob and bring it into where it's fairly good, and then you use the fine adjustment to tailor it, because everybody's eyes are going to be different. You look at something and then you look at something and you're going to have to use the fine adjustment because your eyeballs are different. Everybody's are. So, okay. So this is the microscope. This is a basic compound microscope. There's the ocular, the eyepiece right there. This is called the body tube because light rays that will come up through the objective lens. There's the objective lens right there. Light rays come up through the objective lens. That box that sits on top of the microscope below the body tube, that's a prism. A prism is going to bend the light rays so they go up and then go off to an angle. So they can go up to the body tube, and the body tube then leads to the eyepiece. So light rays come up through the microscope slide, which is sitting on the stage of the microscope, through the objective lens, through the prism. It's bent around and is shot up through the body tube, and then we see it through the ocular. The objectives are held in place on something called the nose piece, and the nose piece rotates it turns around, so we have the choice of picking out which lens we want to use. And the lenses are right there. We have blue, yellow, and red. A 40x objective lens is always blue. The 10x is always yellow, and the 4x is always red. And there we have our scanning, low, and high power. There is our stage. It used to be that you put a slide on a microscope stage and you'd have to move it around with your thumbs. And just when you find something, you'd bump it and you'd lose, a lot of time that you were trying to, when you were looking for something on the slide. Well, these stages are mechanical. We've had mechanical stages for a long time. They allow us to move the slide back and forth or side to side, as well as up and down. So we don't have to touch the slide anymore. And we still make mistakes. Things still happen. And we still bump things when we shouldn't. But it's a whole lot easier to move the slide around when you're looking around. If you're working in blood work and you're looking for a specific type of blood cell, it's a whole lot easier to scan back and forth on a mechanical stage, looking for one of the more less common white blood cells than it is to have to try and push it by hand across here. It used to be you would see someone working in a lab. They'd have a little clicker in their hand, and their eye would be on the eyepiece, and as they would be going back and forth, they'd be counting the number of cells by pressing this little clicker. It was not exactly high tech. I mean, it worked. It's a whole lot simpler now. Most of it is, the scanning is done using technology, but old school was still counting it with a clicker. So I can't imagine how tedious that could have been. So, okay, there's the coarse adjustment, and in this case, there's the fine adjustment there. There's a hard-hitting point of information on this microscope. That's the light. And here's another hard-hitting point here. That's the base. So I've said that I've gone through this slide too many times to take it too seriously. This is the base. The only thing about the base to remember is when you carry the microscope, keep your hand underneath the base, okay? Because they're heavy. They're not that heavy, but they're awkward. And, they fall down. And, what happens if you drop one and it breaks? Well, it broke. Okay, and it has happened over the years, but it doesn't happen very often. So you always carry it like this. Just, I've never had one in any of my classes break, but that's not to say it can't happen. So, and nobody gets upset if it does. So, okay. And there's the light switch. Now, that sounds like a really tedious little point there. We have 3 different types of microscopes. They all turn on and turn off at different places. This one turns off at the back. Others turn off on the side. Some turn off around the big dial. There's no consistency. So when we start using the microscopes next week to look at stuff, just take a few minutes and figure out where your on and off switch is. The other thing you'll want to see is where your dimmer switch is, because these things get really bright and they all have dimmer switches built in so you can lower the intensity of the light coming through. Because there's nothing more exciting than turning it on and looking right in the eyepiece and discover that somebody left it on high. And you're burning out the back of your retina because it's so bright. So there's the iris diaphragm right there. There's a little lever that lets you adjust that. And here's a close-up of all that stuff. There's the mechanical stage again. Let me get past this and get to that next slide. Okay. Now, this is the condenser right there. Condenser works like a magnifying glass. it focuses the light rays that come up through the slide. The slide sits on top of that. The light shines up through there, goes through the slide, it goes through the condenser. It's focused on the slide and it goes right into the opening. That's what the condenser does. It condenses the light rays. And so instead of having the light rays spread out all over the place, they're focused to a point. You know, it is essentially a very small magnifying glass. Those two dials that you see there allow you to move the stage to the left, to the right, or to front and back. And the coarse adjustment and fine adjustment allow you to move it up and down. And that's basically how the stage works. The stage also has a series of clips. You can see part of one right there that hold the slide in place. You know, it used to be that these little clips that you laid on top of it, and you know, you'd mess it up when you're putting it underneath the clip. These are all spring loaded, and you can see it. They just open up, hold the slide in place, and then you're good to go. Okay. And there's the iris diaphragm again. Now, okay, this is a good little slide to remember. It just shows you the magnifications. If the eyepiece is 10, the eyepiece, the ocular is always going to be 10. The scanning lens has 4x magnification. Its total magnification is 40. If you're on the low power, it's 10x, and that gives you a total of 100. And if it's 40x, it gives you 400. So you simply multiply the two together. You know, the ocular is always 10. The ocular is always 10. So it's 10 times whatever lens you're using. And you would be surprised how many people get something that simple wrong on the lab exam. You know, it's sort of like they forget how to do math to multiply simple numbers together on the exam. I guess, you know, I don't know, they get nervous or something, but it's very simple. It's 40, 100, or 400. Okay. Now something else that we see with our microscopes is what's known as resolving power. Resolving power is the ability to differentiate or distinguish fine detail in something. You know, if you look at, if you were to stand in the parking lot at night at the far end of the parking lot and hold up 2 matches or 2 lighters, doesn't matter, and have someone on the other side of the parking lot, they're all the way, you're all the way down in front of ACAD, and someone else is in front of CAPE, and you hold these 2 matches together like this, or 2 lighters, whatever, and they're both burning. Now, from 1,000 feet away, it looks like 1 light. From 500 feet away, it looks like 1 light. If you have a pair of binoculars that you can zoom in on what the person's doing, you can see, no, it's not 1 light, it's 2 separate lights. That's resolving power. Resolving power allows us to see differences in structure that we can't determine any other way. Consider this. This is a letter E. And I'll get into the part about being upside down and backwards. Now, when you have something printed out, you know, if you had a handout, and you know, it's more like the attendance sheet, for example, and all the names are on there, and there's a whole bunch of letter E's in everybody's names. We look at it. It looks very sharp and crisp. It looks very nice and clean, straightforward, you know, nice sharp edges. But when we magnify it, a letter E looks like this. It looks like has the fuzzy edges there of the splatter of the ink on the paper. Our eyeballs can't see those edges. You know, we're looking at something that is, you know, 12 point, you know, or even 20 point, or even if we go all the way to 72 and print it out as big as we can get it, we can't see the rough edges where the ink splattered on the paper or the toner splattered on the paper. If we magnify it to, to like 100 times or 200 times, then we would see this. Resolution is the ability to distinguish irregularities in a surface or on an object or on a structure. Our eyes don't have that ability. We can't magnify that much. So, something else about the microscope is that everything that you see is upside down and backwards because of the nature of the lenses. And it doesn't matter, because for a bacteria or for a cell, there is no upside down or backwards. Doesn't, we're not going to do a whole lot of reading through the microscope to look at letters. But we will see, so when we see a cell, it doesn't matter that it's upside down and backwards. How do we know that how do we know it's upside down? How do we know it's backwards? So we're just looking at the image. Something else about looking at in the microscope is that you're going to see different cells and tissues have different colors because we stain them. Because if we look at human tissues, they all look the same. Most of our tissues are going to have the same appearance. They're going to be pale and have very little color to them. Muscles tend to be darker red because of the presence of myoglobin. Myoglobin is a pigment in the muscles. So the heart and the skeletal muscle and the smooth muscle all have the same darker appearance. The liver has the same darker appearance, but all the other tissues in the body tend to be just sort of a washed out pale coloration. So we stain them so we can see things. So we're going to see tissues that some look purple and some look pink and some look bright red and some don't have any coloration whatsoever. Some are going to look blue depending on the dye, the stain. We use a stain so that we can at least differentiate between the background and the structure. It gives us contrast. There are a variety of stains. Now, next week when we do this thing called a wet mount, we're going to use something called methylene blue as a stain, and it stains things blue. And so if we look at cheek cells do very well with methylene blue. Now, our cheek cells are not blue, unless we're cyanotic. But these cheek cells pick up the methylene blue. It shows you the nucleus, it shows you the cytoplasm, and it shows you the edge of the membrane of the cell. That's about all we can see with these microscopes anyway. Without the dye, we might be able to see the nucleus, but we probably wouldn't because the whole thing's transparent. So you don't see things very well unless we use a stain. And there's a wide variety of stains that we use, particularly when we get into bacteria. There's all sorts of stains. When we look at different types of bacteria, they can be stained purple and pink, and some can be bright yellow and some can be bright red when we look at them. So there's all sorts of categories. If you take microbiology, you'll get into that. It's different things, different stains for different tissues, if you will. So, okay. And this is what it looks like if you do a real cheek cell. Here's another one right there. Okay. Now, thank you for bearing with me as I go through that. Let's move on to the next part of this. And let's get into, okay, epithelial tissues. Okay. Now, in the human body, there are we only have 4 types of tissues. There are only 4 categories of tissues. Epithelial is probably the #1 tissue type. Epithelial cells cover all the openings to cover the surface of our body and cover all the openings into our bodies. So epithelial cells line our digestive system from the mouth to the anus. They line our reproductive tract. They line the inside of our ears, and they cover our skin and the inside of our mouth and any opening, our urethra, the inside of our nose, the tissue around our eyeballs, anything that's open, any area in our body that's open to the outside. And we consider the reproductive tract, the urinary tract, and the digestive tract open to the outside, because it is. We consider mouth to the anus is a 20-some foot long tube. Yes, it has to go through the esophagus and the stomach and the small and large intestine to get to the outside, but it's still an open tube straight through. So that's all lined with epithelial cells. And epithelial cells are very specialized for us. They're there, well, among other things, they're there to protect us. They're our first line of defense against pathogens, bacteria and viruses. They form an impenetrable waterproof surface on the skin. At the same time, they also become a passageway to absorb nutrients in our small intestine. Same type of tissue, epithelial cells. They can be a barrier, they can be a passageway. And, as I said, there's only 4 types of tissues in the whole body. Epithelial is one of the 4 types. Now, within the epithelial cells, there's only 2 categories: simple or striated. Simple means 1 cell layer thick. Simple, an example of a simple epithelial cell are the cells that make up our lungs. The lungs are designed for gas exchange. They are big inflatable structures in our chest that are made up of these specialized bubbles called alveoli. The alveoli inflate when we breathe in air. The alveoli are as thick as a single sheet of wet single-ply toilet paper, and that's probably too thick. They are 1 cell layer thick, so they can expand and contract and fill up with gases. So it is amazing. Simple epithelial cells designed to expand, to allow air to come in and inflate, and allow that oxygen then to pass through the cell membrane of the alveoli into a blood vessel right beside it. It's it's an amazing concept that you have this super thin sheet of epithelial cells that oxygen can just pass right through and be picked up by blood vessels on the other side. Extraordinarily thin, and yet it functions for us all the time. If we were to take a lung and spread out all the alveoli on a flat surface, the surface area of 1 lung would cover the floor in this room. So it's a huge amount. And we have 2 lungs. Now, so simple. Anywhere we want to have movement across the membrane, we're going to have simple epithelial cells, 1 cell layer thick. Stratified epithelial cells are where we have multiple layers. Our skin is stratified because we do dumb things to our skin. We injure ourselves, or our dogs help to injure ourselves for us when they play. And so we have multiple layers of epithelial cells, we call it stratified layers, designed to wear away, designed to be injured, designed to be torn off, designed to wear off, because we're always shedding skin cells. We're always shedding skin cells. You know, when we get injured, we don't bleed until we get down into the dermis of our skin. The epidermis is the outer layer of our skin. The epidermis is mostly dead cells, and with a thin layer of living cells that's sort of the bottom of the epidermis or the top of the dermis. We don't bleed until we get into the dermis. So, because we're always shedding off some of these stratified layers are designed to protect us. Our mouth is lined with stratified epithelial cells. Our entire digestive system is lined with stratified cells where we're going to have wear and tear. Our mouth is lined with these cells because eating causes a lot of blunt force trauma to the inside of our mouth, depending on what we're eating. You know, you're eating an apple. Well, you're a lot of chewing going on there and it's tearing away cells. What about popcorn? Or what about something, a pretzel, something that's going to get, or chips, something that's going to tear away more cells. Well, it doesn't hurt to eat, thank goodness. But it is going to tear away the cells in there. So we have lots and lots of layers to protect us. But now let's move down below the stomach into the small intestine where we absorb nutrients. We can't have multiple layers there because we won't be able to absorb nutrients. We get down to single layers there. But then we move a little further down into the colon, we get down to the rectum, and we're passing fecal matter out. There again, we have multiple layers because that's a lot of wear and tear, on the colon when we defecate, we'd be very unhappy if there was a lot of pain involved in defecation. So anyway, we have a sore butt all the time, I guess you say it like that. So, okay. Now, simple one cell layer thick, stratified, multiple layers. There's only 3 types of epithelial cells, what we call squamous epithelial. They are flat and irregular. They've often been described as fried eggs. I don't know why. I guess because the nucleus sticks up in the middle like a fried egg. Cuboidal are shaped like little cubes, and columnar are shaped like little columns. So those are the only 3. Those are the 3 types of epithelial cells. We have another type called transitional, which stretch. They look like squamous. They look like, let me rephrase that. They look like cuboidal, but they stretch out like squamous. And then we have one called pseudostratified ciliated columnar epithelial, which are really unusual looking. I'll show you those here in a second. Okay, these are squamous epithelial cells. Simple squamous. This is what makes up our lungs. The alveoli, the gas exchange site in our lungs, the one cell layer thick, because it says simple, the one cell layer thick structures in our lungs, this are simple squamous epithelium. If we looked at it from the side, you'd see the nucleus sticking up like where we get the fried egg analogy. So thinner, single ply wet toilet paper is about the best I can come up with as an analogy compared to this. But there is simple squamous epithelium. Here we see simple cuboidal. They look like cubes. That's why they're called cuboidal. We see these in our kidneys. Our kidneys are filled with tubules. What you're looking at here in the center of the screen, where you see all these cube-shaped cells right in the middle there, you're looking down into a kidney tubule from the top. And the filtration process of filtering our bloods 60 times a day. Every 45 minutes, our whole blood supply is filtered. But if you consider how many times a day that we filter our blood, The filtration is taking place through the walls of these tubules from side to side. And they are also one cell layer thick. There's a better shot of the simple cuboidals there. Again, at the kidney tubules, the kidneys are about the only place where we see these simple cuboidal cells. These are simple columnar cells. They look like columns. We find them in our small intestine. We find them anywhere we want to absorb or secrete nutrients or materials, because nutrients in the small intestine are going to pass through these simple columnar cells and pass into the capillaries underneath them. And so we absorb our nutrients. They're shaped like little columns. That's why they're called simple columnar, one cell layer thick. Now, what we also see here, this is what's known as a goblet cell. These little white structures here, these goblet cells secrete mucus. We lubricate everything. We don't like to have food pass through our digestive system unlubricated. We secrete mucus so that it doesn't hurt. We secrete mucus in our esophagus so that food passing down the esophagus doesn't hurt. We secrete mucus in our small intestine and our large intestine. We secrete mucus at the rectum and the anus so it doesn't hurt. Anywhere we want to have a passage of something, we're going to secrete mucus to lubricate. And that's what these goblet cells do for us. There's another picture of a better picture of your simple columnars with a lot of goblet cells here. This is a portion of, again, a portion of the small intestine. Somewhere back in here are some capillaries that are going to absorb the nutrients that have passed through the simple columnar cells. Again, goblet cell there. Now, this one is the pseudostratified ciliated columnar epithelial cells. It's a mouthful to say that. Where do we find them? We find them in places where we are trying to move mucus or materials against the flow of the contraction of the structure, if you will. So for example, our airway is lined with pseudostratified ciliated columnar epithelial cells. Now, the reason that they're there is because our airway, our trachea, is covered with mucus. You can even see there are the goblet cells. There's cilia. Oh, let me get back one. There's a goblet cell. There's a goblet cell. See, when we breathe in air, it's got all sorts of crud in it, all sorts of dust and debris and bacteria and pathogens and foreign objects and things like that. And we suck that in. That the cilia that you see there, cilia are little tiny hairs, and they move. They move in one direction only. They move forward and they rebound and do it again. They're covered with mucus, the mucus from the goblet cells. All that dust and dirt and crud and debris that we suck in when we breathe gets trapped in that mucus, so it doesn't go down into our lungs. The cilia then push the mucus up to our throat, and we swallow it. And we swallow about a pint of that mucus every day over a 24-hour period. Now, we don't notice it. We're not aware of it, but we swallow it. It goes into the stomach and gets destroyed by the stomach acid. So we get rid of the invaders, if you will. That's what the cilia does. It's constantly beating, bringing the mucus up. Unless, of course, your patient's a smoker. Tobacco smoke, or rather to say cigarette smoke of some type or another, paralyzes the cilia. And so it doesn't move the mucus out. A smoker, excuse me, will get up in the morning and start coughing. the smoker's cough. You ever heard the term smoker's cough? A smoker will start coughing the first thing in the morning to try and cough up a lot of this mucus because the cilia are paralyzed by the chemicals in the smoke, and they can't do their job. They can't move. Now, the good news is if a person is a smoker and it stops, then the cilia eventually come back and continue to work again and get rid of the mucus every day. Prior to, but when they're paralyzed, they can't. So that your patient would cough to get rid of the mucus. So, and here's a better picture of it. The reason we call it pseudostratified is it looks like they're multiple layers. They're not. They're a single layer, but they look like it's a multi-layer. But there's the cilia on top. We see this in the trachea. We see it in the fallopian tube in the female. When an egg is released from the ovary and drifts into the fallopian tube, the egg has no way to get down to the uterus on its own. The egg doesn't have a tail like sperm cells do. The egg will just have to drift. Now, to assist the egg in getting to the uterus, the oviduct, the fallopian tube, is lined with pseudostratified ciliated columnar epithelial cells that will push that egg along. Doesn't matter whether it's fertilized or not, but they'll push the egg along. Now, if it's not fertilized, the egg won't implant. In many cases, the egg doesn't implant anyway. So, but this is what the cilia, and this is what they look like. This is what they do. They line the oviduct, the fallopian tube, and drag that egg along to get it, at least to get it to the uterus. So it's just a few places in our bodies where we're going to have cilia. Everybody's got them in their lungs. It also shows up in the oviduct. Okay. Now this other one here is a bit of an, this is a really poor picture. I'll just tell you that. You'll see a better picture later on. But this is a slide that represents, this is a portion of the urinary bladder. And urinary bladder is our urinary bladders are pretty unique, is a pretty unique organ. It can, at complete rest, it's about the size of a walnut, and it can stretch out and expand to something the size of a grapefruit. It can go from holding a couple hundred milliliters of urine to well over a liter of urine. And it expands. Now, transitional epithelium has the ability, there we go, transitional epithelium has the ability to stretch out as the bladder expands. And when it expands, it looks, it starts out looking like columnar. See how it looks like little columns? And when it stretches out, it looks like squamous. It goes from being columns to flat and then back down to columns. Because it expands, we empty our bladder, it shrinks back. It fills up, we empty our bladder, it shrinks back. So we have this transitional, because it has the appearance of two different types of epithelial cells. Okay, this is transitional epithelium here. Again, where do we see it? In the urinary bladder and in the urethras and in the ureters, I'm sorry, and part of the urethra as it stretches. Our bladders can only hold so much though. You get above a liter of urine in the bladder, and the bladder, it can rupture if you hold it long enough. But usually, it forces you to urinate at some point long before that happens. But it's still possible. So, and okay, and this is stratified epithelial cells. Just layer upon layer upon layer of squamous epithelial. This is what lines the inside of our mouth. This is what lines our rectum. This is what lines the uterus. This is what lines the urethra. This is what lines the esophagus. So anywhere we're going to have wear, and this is what makes up our skin. Anywhere where we're going to have wear and tear and have skin cells torn away all the time, and we're not aware of it, you're going to have this stratified squamous epithelial cells here, busy doing their thing. So, okay, now, and of course, there's, again, the esophagus, the mouth, the vagina, the urethra, the anus. It may or may not have goblet cells. Surface of our skin is going to be waterproofed. We certainly wouldn't want to have our rectum and our anus be waterproofed like sandpaper. That would be painful. So same type of cell, it just depends on where it's located. Okay. And that is the end of epithelial. And I think I've probably bored you to tears. So I think I'm gonna, would you like to call it a day? Okay, that's good. I do too. So let me stop the sharing. Yeah, we've covered a lot today. I always think, well, I can go through all the tissue types in one day and everybody will be fine with that. It's like after an hour of this, it's like, no, I can't go any further. So, okay. Next week in lab, we'll wrap up the rest of the tissues. We'll have the experiment we're going to do about looking at movement across the membrane. We're going to create an artificial cell and we're going to measure the movement of fluids across the membrane. And that takes about an hour and a half to run. And while that's running, we can finish talking about this other stuff and maybe make some cheek cells slides and prove that we're all alive. So. Anybody have any questions on anything? Okay, then I'm going to get us out of here and I'll see you all at lecture later in the week.